So hello and welcome back. So with the continuation of the cell biology, human cell biology, we'll be discussing the cell theory. This theory was proposed together by Sladen and Swan in year 1839 AD. So how the cell theory started? The cell theory started when two German biologists, botanist Matthias Sladen and a zoologist started independently observing cells. So Sladen was a botanist who saw the cell and the entity in plants, whereas Swan was a zoologist and he saw the same thing in animals. Only the thing he found different was animal lacked a cell wall. So Swan gave an hypothesis called as cell hypothesis, and Sladen and Swan came together, published a joint paper in year 1839 by the name of cell theory. And in that, he proposed these basic fundamental theories that all the living organisms are composed of cell and their cell product. Means all the living organisms, what we are seeing here, internally they are made up of cell only. And what the cell is made up of a small mass of protoplasm with a nucleus inside and plasma membrane outside, with or without a cell wall on the outside. So when Sladen proposed in the plant, he said with a cell wall, and when Swan saw it, he saw it without a cell wall because he saw an animal cell. So this term with or without cell wall came into existence. And the third point was all the cells are basically alike in their chemistry and physiology. Means if you zoom into the cell, you see the protoplasm, and inside that there is a cytoplasm. Cytoplasm has the same pH. The mitochondria of all these cells perform the same basic function. The ribosome also has the same function in all these type of cell. And from there he saw and he proposed that basically all the cells are alike, no matter if it is a cell from the blood or it is a cell from the intestine or from the liver cell or from kidney. All the cell, when you zoom in, you see the similar entity of structures and also the similar type of functions in them. And from here he proposed this third point, all cells are basically alike in their chemistry and physiology. And fourth one is the most popular one that we have already discussed in our previous video, that activity of an organism, either the digestion or movement or anything, any other activity, is the sum of total activity and interaction of its constituent cell. So this was the basic of the cell theory he proposed, and this is very important one. One should remember these 4 points. And the person who have proposed this theory. So after the cell theory was not just halted here, but sooner after the cell theory, many discoveries were made. Like Robert Brown, there was a scientist named Robert Brown who came up with the idea of a nucleus. He discovered the nucleus, by the way. Other scientists came, and in that one, the common or the most popular one was Rudolf Virchow. Rudolf Virchow said that a cell exists from only by division of the pre-existing cell. The cell don't come up from the air like snap. A cell only comes from a pre-existing cell. So this is a very popular finding of his. He proposed this phrase called omnicellula is cellula. A cell derives only from a pre-existing cell. So finding like of the Robert Brown, Rudolf Virchow and others, many findings contributed into the cell theory, and cell theory finally was updated again and again. Means more points were added into the cell theory. And this cell theory was not limited only to the Sladen and Swan original cell theory, but became something new. And the new cell theory was known as modern cell theory, which was contributed by many other scientists, and their discovery backed up this modern cell theory. So this modern cell theory proposed many points on the cell theory, and the most common or the most important ones are cellular composition. Here it says that all organisms are composed of one or more cells. This means all the organisms what we are seeing right now, or what it is, all the organisms what we are seeing or cannot be seen, these all are made up of cells and their product. So second one is a structural unit. This says cell are the fundamental building block. So whatever the body or the bigger of a organism or animal or plant or bacteria or fungi, anything is there, all of them are structurally made up of cell only. Not just structurally but functionally also they are made up of cells. Their working is contributed by cells. Third point was cell division. Omnis celluli cellula, which means new cell originate only from pre-existing cells. Another point was energy flow, where inside the cell there are multiple organelles which transfers its material to the another organelle and finally from another organelle to another organelles, and that's how energy and product flow inside the cell organelle, and finally the cell performs its specific function. Like for digestion, if the pancreas starts producing enzyme, then it is the cell inside the pancreas which is actually producing the enzyme, the pancreatic acinous cell, and inside the pancreatic acinous cell the organelles are that one which are producing that enzyme. Like the ribosome and on the rough endoplasmic reticulum is producing that enzyme, and that enzyme reaches to the cell membrane, and from the cell membrane it pops out from the pancreatic cell, and it finally reaches into the intestine where it performs its specific function. So it tends to say that inside the cell, this energy flow point says that inside the cell, the organelles are the one whose interaction performs a physiological action. Another point is genetic material. Cell passes on its genetic information during cell division, and the detail of it will sooner follow in another upcoming videos in a detail. So chemical similarity, all the cells are similar in chemical composition. So collective action. Organism activity is the result of cellular interaction. I'm going to give you a very good example. Suppose a person decides to run. So for this, brain produce signal. That signal is produced by neuron. That neuron signal transmit to the spinal cord. That is again a neuron, made up of neurons. And that finally reaches from nerves, N-E-R-V-E-S, and from the nerves it reaches to the neuromuscular junction and here to the muscle, and inside the muscle there occurs contraction, and when the muscle contracts, muscle attached to the tendon, and that tendon is attached to the bone, and when the muscle contracts, bone also moves, and that movement of bone produces a mechanical movement of the body. So this running is contributed by the neuron and muscle. So two different type of cell contribute to the running. Other things like one which provides the energy, like the liver is providing the energy, the blood cell are providing the energy so that the muscle contracts and relax in a regular manner so that the person can run continuously. Other cells are also contributing in that process of running. So this action of running is the collective action of more than one cells, like neuron, muscle, the liver cells, and the RBCs and cells as such, and finally the person can run. That is a good example of this collective action. Talking about totipotency, zygote is formed after fertilization, and that zygote finally forms a baby. And that baby grows up into an adult. So fertilization involves two haploid cells which fuse to form a single cell or a zygote, and that cell divides continuously from a baby, and finally that baby grown into adult. The adult has many systems in a body, like a respiratory system, excretory system, skeletal system, musculoskeletal system, nervous system, and system as such the adult person had. So isn't that wonderful that a single cell divided so many times to form different type of cell in the body of an adult, like trillions of cell inside the body of adult, and the cells are not just present in a lump inside the adult, but they are differentiated in different activity. They contributed to form different system inside the body. Just this single cell did all this thing inside the adult. What we are here we are seeing, it is a contribution of that single cell only. So this what the totipotency means. A single cell has ability to differentiate into multiple cell and into multiple type of cell, not just a single type, but multiple cell and of multiple types to finally form different organized system. That ability of a cell, that potency of a cell is called as totipotency. So another point is common ancestry. So this point again proves the common ancestry. All the cell shares common evolutionary origin. Nervous cell, excretory cell, reproductive cells, any type of cell, all cell was developed from this zygote. So that's what it means. All these cells shares a single common ancestry. Now we'll be discussing about type of cell and parts of a cell and internal structures of a cell. So describing the type of cell, here are two types of cell. First one is prokaryotic cell and second one is eukaryotic cell. So how this prokaryotic cell and eukaryotic cell are divided into types? So this division is based on the nucleus, cell organelles, the cell wall and cell membrane. So based on these four parameters, the cell are divided into prokaryote cell and eukaryote cell. So in the prokaryote cell, you can see here, there is not a prominent nucleus or any nuclear membrane inside it. So the chromatin body of the DNA is directly scattered into the cytoplasm, and such a condition or such a type of nucleus is called incipient nucleus. Incipient means it is old or primitive, yet to be formed. Means it is not a complete nucleus or a completely formed nucleus or completely mature nucleus. It is just primitive early nucleus. That's what incipient nucleus means. Here, this chromatin is lying directly scattered into the cytoplasm, and the genetic material is the DNA, same as we do. It is of circular type, however. We have linear DNA, but this one is circular. There is no histone in it. There is no histone protein in bacteria. Now, talking about the cell organelles, no membrane bound cell organelles. This means no mitochondria, no endoplasmic reticulum or Golgi body here. Things like that with the membrane bound, they are absent in the bacteria or the prokaryotic cell. Prokaryotic cell means bacterial cell we are talking about. It also includes blue-green algae. Now, the cell wall, here I haven't drawn the cell wall, but understand this. This cell wall is covering all over the bacterial body. The cell wall is richly made up of lipid and protein, or richly made up of the peptidoglycan type cell wall is in gram-positive bacteria, and lipid and protein rich cell type cell wall, is a gram-negative bacteria. So, but in case of a cell wall of the eukaryote cell, it is made of cellulose, hemicellulose and pectin, which is different from that of prokaryotic cell. So, cell wall is different. And coming to the cell membrane, there are dissimilarities also. In the bacterial cell membrane, there is a lack of cholesterol, sterols and complex lipid molecules in them. However, inside human cell membrane, there are presence of complex cholesterol, sterols. So that's how the cell membrane is different when the prokaryotic cells and eukaryotic cell are compared. Yeah, about cell organelle also, ribosome type is also different. Here in us, there is 80 S type ribosome, but in bacteria there is 70 S type of ribosome. That S means and the type of ribosome will also come up in detail later. We will again discuss this. Eukaryote on development formed several differentiations, and that differentiations led to the classification of eukaryote cell into plant cell, animal cell, fungal cell and protist. Protist means unicellular organisms. We have discussed in previous video about cell that has independent existence, unicellular cells like parameciums. In the upcoming details, we will be focused mostly on this animal cell wall because we are dealing with or studying the human cell biology, which is a part of an animal cell. Yeah, also I forgot to tell you, in between them also there are some differences, like protist is unicellular, made up of single cell wall only. Fungal cell wall has a difference compared to these two or others also. The fungal cell wall contain ergosterol and cell wall has chitin in it. The animal cell and plant cell wall are differentiating based on their upper covering of the cell, that is the cell wall which is present in the plant cell and it is absent in the animal cell, and also plastids and food preparatory structures and mechanism are present in the plant cell which is absent in the animal cell. The detail of the difference between prokaryote and the eukaryote, the plant cell, animal cell, and all are kept in the Google Drive. The links will be sent in the description. Now here we are looking at an unlabeled animal cell. Let us start looking into the structures and property and properly labeling them as well. So let's start from the surface. So this membrane like a structure is cell membrane. Cell membrane is the outermost covering of an animal cell which separates the external environment and the internal cellular environment. It leads to the compartmentalization of a cell. So beneath the cell membrane, all the part here is known as cytoplasm. In the cytoplasm, the clear liquid-like part, whatever the matrix, all this clear part here without any organelles is called cytosol, and another part is organelles and bodies inside the cytoplasm. Let's start discussing about the bodies inside cytoplasm. So the central is nucleus, and the nucleus is externally surrounded. It is surrounded by a double membranous structure called nuclear membrane. This membrane tends to separate the inner nuclear structures and the nucleoplasm from external cytoplasm, this nuclear membrane. And after that, the nuclear membrane is continuous here. See here, it is continuous with a external structure, and that structure is endoplasmic reticulum. To be precise, it is rough endoplasmic reticulum. Now, here you can see double membrane bound structures lying in the cytoplasm. These are mitochondria. Mitochondria the same, the powerhouse of a cell. These structures what you are seeing here are Golgi bodies. Apart from rough endoplasmic reticulum, there are other endoplasmic reticulum also called smooth endoplasmic reticulum, which lies just after the rough endoplasmic reticulum. And this is this. So I'll better draw it like this. Now these structures are lysosomes. They are enzyme-filled sacs which perform digestions inside the cell. These are vacuoles inside the cell. These are cytoskeleton. In cytoskeleton, it can be microtubules, microfilaments, anything. So just label this as cytoskeleton is scattered in the cytoplasm. They maintain the shape of a cell, integrity of a cell, and also tends to connect organelle and keep the organelles in its own places. So it's like rods inside the cell that connects and keep the integrity of the cell together. Other structures are centriole. In centriole, it is only shown one here, but on the back side of it also there are another one similar to this, lying like this at 90-degree angle. The detail will come up sooner. And this is small dot-like structure you are seeing here. These dots are ribosomes. Yeah, also about this rough endoplasmic reticulum, I did forget to tell you. These rough endoplasmic reticulums are not present only by itself, but they are also coated by this small granular structure of ribosomes. That's why the ribosome can look like this, its texture as rough. So that's why it is called rough endoplasmic reticulum. It is coated by the ribosome. These small hair-like projections coming out of this animal cell are microvilli or membrane projections. So if it is in a small intestine, this type of projections are present in the absorptive surface, which increase the absorptive surface of a cell, that is a microvilli. But if it is present in a respiratory epithelium, then it can be cilia, which tend to perform the sweeping motion to sweep away the dust and foreign particles. And also in unicellular organism, these cilia are responsible for motility. A structure like these now and these are responsible for attachment of a cell to another cell so that all the cell can stay together and work firmly. These structures are called desmosomes. Desmosomes forms tight junction, gap junction and junction like that tend to connect more of the animal cell with another cell, form a compact and sturdy tissue. These invaginations you can see here, this invagination occurs or forms inside the cell when any food particle is about to enter here. The food particle are collected here and finally this membrane connects to this membrane and finally this will pinch in and it will form a food vacuole. So these are phagocytic or pinocytic vacuole. These are, if solid food is present then it is phagocytic. So it is not a vacuole yet, so we call it as a phagocytic or pinocytic structure or phagocytic or pinocytic invagination of a cell membrane. This is the detail about animal cell. Apart from this, there are other structures also which is not listed here, like glycosome, peroxisomes, etc., which will come up eventually in the next slides or in next videos.